Okay. So when we're talking about metabolism, really, that could refer to a couple different things, but when we're talking about it in this class, we really just mean cellular respiration. So we're trying to figure out where ATP comes from in a cell, which for our purposes, when we're talking about cellular respiration, it's really from the breakdown of glucose. So glucose, one of our monosaccharides. ATP is a nucleotide that basically has 3 phosphate groups on it. So we can build ATP by tacking on an extra phosphate group to the same molecule when it only has 2 phosphate groups. So we'll start with a molecule called ADP, adenosine diphosphate, and then we'll tack on another phosphate group, put energy into this bond to make it adenosine triphosphate. And when we eventually use ATP, we'll see that we just do this in the reverse. When we pull off the final phosphate group, that gives us energy that was stored in that bond. So this is an endergonic process. We can break down ATP. We talked about this on Friday. So moving into actual cellular respiration, we talked about the fact that we have a couple different steps. They create ATP in 2 different ways. So we have substrate level phosphorylation, where we use an enzyme to bind on this extra phosphate group. We get it from some other molecule. And then we have oxidative phosphorylation, which works a little differently, using something called the electron transport chain and a gradient of molecules across a membrane. So glycolysis, one of our substrate level phosphorylation methods, produces 2 ATP on net. So we put in 2 molecules of ATP, in an energy investment phase. I know I'm going through this fast, but this should all be reviewed. So we put in 2 molecules of ATP to destabilize the molecule, basically. This destabilized form is called fructose 1,6-biphosphate. We then can split the molecule in half. This gives us 2 pyruvate molecules. I hate that that pops up every time I point to the bottom of the slide. Back the pointer on, does it still do it? Yeah, inconvenient. Okay, so from glucose, we end up with ultimately 2 molecules of pyruvate. We also end up with our 2 ATP on net. We also end up with this molecule NADH, which is something called an electron carrier. So we'll see it come back later in the process. The actual production of ATP happens in this energy harvesting phase. So this is after we've split our glucose molecule in 2. Okay, so this is where we left off. We can see that lots and lots of different types of enzymes catalyze the steps in glycolysis and the steps in the rest of cellular respiration. We're not concerned about their specific names for this course, but we saw this word kinase before, for example. So you can see that there's an enzyme here catalyzing this conversion of ADP to ATP. That's one of the places we're performing this substrate level phosphorylation. We can see this phosphate group that we grabbed here as well. Okay, so that's substrate level phosphorylation. Okay, so after glycolysis, we have a next step. Here, for the purposes of this class, we're going to think of this as a linking step rather than a named step on its own. You may have named this step out as its whole own section of cellular respiration in previous classes. So this linking step, we're taking the pyruvate molecules. We have 2 pyruvate molecules at the end of glycolysis. We're moving them into the mitochondria, and we're converting them to acetyl-CoA. Okay. In this process, we also create more of our electron carrier here, this reduced NADH, and we release some carbon dioxide. So one of the main byproducts of cellular respiration is carbon dioxide. This is why you breathe in oxygen and breathe out carbon dioxide, is you need the oxygen to break down the glucose. And then all those carbons in the glucose, you don't actually need those carbons for the ATP, so you breathe them out. So we're going to produce CO2 throughout this process to get rid of that piece of our glucose molecule. We really just want the energy in the bonds of the glucose molecule rather than any of the actual elements from it. Okay, so this is our linking step, and it's linking because we're moving to where we want to go, basically. So our linking step, we have pyruvate, and we have this additional molecule, coenzyme A, that's put into this process. And we need to put in our precursor to our NADH, so we put in some NAD here. And we get out, ultimately, this acetyl-CoA. That's our main product that's going to stay in this pathway, stay in cellular respiration. Okay. So acetyl-CoA leads us into the Krebs cycle, also known as the citric acid cycle, also known as the TCA cycle. So good, they had to name it 3 times. Great. Okay. So the Krebs cycle is a cyclical metabolic pathway, which is to say it has a number of steps and they all lead back to the first one. So this can just keep going round and round and round. And we're just going to be giving stuff off kind of on the sides of this cycle. The Krebs cycle is happening inside the mitochondria. So during our linking step, as we were breaking down our pyruvate to make our acetyl-CoA, we moved our products into the mitochondria. So now we're inside the mitochondria. Very important to remember that glycolysis was occurring outside the mitochondria, but now we're inside the mitochondria. And that's important because when we start thinking about what happens when we don't have oxygen, we'll come back to that. Okay. So the Krebs cycle is also the next place that will create ATP. So if you paid careful attention to that equation about the linking step, we didn't produce any ATP there. We're just linking together glycolysis and the Krebs cycle. We're giving off some waste products, but we didn't actually make any of this main molecule that we're trying to track. In the Krebs cycle, we do though. So we can see here, the bottom of the cycle, we're producing 1 molecule of ATP. It's important to realize that we're actually going to basically go through 2 rounds of the Krebs cycle for every molecule of glucose, because we had this cleavage step, this splitting step where we split glucose in half. So everything after glycolysis basically happens twice. Okay, so we're inside the mitochondria, we have this cycle, we're reducing more coenzymes. Now that's really the main purpose of the Krebs cycle. So we do produce this tiny amount of ATP, but because the Krebs cycle is going to lead us directly into our next step, our electron transport chain and our oxidative phosphorylation, which makes a ton of ATP, that's not that important here. But what is really important is this electron carrier. So our NAD or NADH is a coenzyme. So our reduced form here is the NADH. And this is really important to the function of the electron transport chain and therefore to oxidative phosphorylation, which is the way we make a lot of ATP using the electron transport chain. Yeah. So that's really what I want you to focus on in your mind when you think about the Krebs cycle, is that this step is really important. We're basically collecting these electrons, saving them up for the next step. Okay. So our initial substrate, what we started with going into the Krebs cycle, is acetyl-CoA. It's immediately funneled into this cycle. There are a number of steps. We don't need to know them specifically for this course. But for each acetyl-CoA, we're producing 1 molecule of ATP. We're producing 3 of this molecule NADH, 1 of this molecule FADH2, which works the same as NADH. Those are interchangeable in function, I would say. And we're also producing CO2. So at this point, we actually get rid of all our carbon from our glucose molecule. There's going to be none left after the Krebs cycle because we got rid of some as we were doing the linking step, as we were breaking down pyruvate. Now we're getting rid of the rest of it as we move through this cycle. Okay. So if we wanted an equation for this, we can see it at the bottom of the slide. So the things to focus on here are the fact that we're producing all these electron carriers. Big picture here with all our enzymes drawn in. And I will save that for your chemistry classes. I guess you have already had your chemistry classes, so hopefully you've gone into this in a little more detail. But what we need to know here is our electron carriers and a little bit about our ATP. Our final step of cellular respiration is oxidative phosphorylation. So in oxidative phosphorylation, this is where we're making most of our ATP. So by the time we get to this step, we've made a little bit in glycolysis. We've made a little bit in the Krebs cycle. We've only made 4 ATP molecules total. So 2 of those were from glycolysis. 1 was from our first round of the Krebs cycle, and 1 was from our second round of the Krebs cycle. So that's how we get those 4 ATP molecules going into oxidative phosphorylation. But all the rest, and we saw that we can make up to 38 ATP molecules through this whole process, is going to be made by the electron transport chain through oxidative phosphorylation. And the way we're going to power this production of ATP is through the use of a chemical gradient. And we're going to see that we're going to have electrons moving across the electron transport chain using that chemical gradient in order to synthesize ATP. Now, very importantly, oxidative phosphorylation, this final step, the electron transport chain, our method of doing oxidative phosphorylation, requires oxygen. Oxygen actually has not been an input of any of our steps up until here. The only place you need oxidation, the only place you need oxygen is during oxidative phosphorylation. Because all that ATP is made by oxidative phosphorylation, that oxygen is really important. So we are able to make a tiny bit of ATP without oxygen because glycolysis can make ATP, because we can do that without any oxygen whatsoever. But in order to produce efficient numbers of ATP molecules, we need oxygen because it's crucial to this step. Okay. So the way this whole system works is that we have this electron transport system, which is a chain of molecules embedded in the inner mitochondrial membrane. So if you remember what a mitochondrion looks like, right, the powerhouse of the cell, this is why it's the powerhouse, it's making all our ATP. It has an outer membrane, so it looks kind of like a kidney bean, right? And then it has this wavy membrane on the inside, making sort of a separate pocket inside the mitochondrion so that we can create chemical gradients in the very center of the mitochondria versus this sort of intermembrane space, so the space between the 2 membranes of the mitochondria. Okay, so we have this chain of molecules in the inner membrane that are going to undergo a couple of oxidation reduction reactions going from high to low energy. So as we move down this chain of molecules, we're going from high to low energy, but we're releasing more and more energy for this process. The energy we release from the electron transport chain is used to synthesize ATP by this process of oxidative phosphorylation. So when you see the term oxidative phosphorylation, we're referring very specifically to this process going on here. Doesn't happen during any of our other steps. Okay. Our final molecule that we want to concentrate at the end of the electron transport chain, after we've done these oxidation reduction reactions, is ATP synthase, which is going to do the actual production of the ATP. But as we're going down this chain, we're basically like passing along electrons down a chain, and our final electron acceptor is oxygen. And that's why we need the oxygen at all, is because it's the end of this pathway. So if we don't have oxygen, everything gets backed up. We can't pass all the way down the chain. So eventually the chain just stops running because we never get to this final step of producing ATP. We're going to look at this a little bit in more detail at how the electrons are donated to the chain. Okay. So the electrons that we're using in the electron transport chain, right? Electron transport chain is transporting electrons, and it's basically getting them from our electron carriers. So the reason we were reducing NAD to NADH here is that we were like storing these electrons to take them to the electron transport chain. So most of them came from the Krebs cycle, but some of them came from other places, earlier steps as well. Now our final accepting of these electrons is happening through oxygen. So our final place that these electrons go is to our oxygen molecules, this air that we're breathing in. So when this happens, we bind together 2 molecules of hydrogen ions plus some of our oxygen to get water. So that in addition to the CO2 we're breathing out, we're also going to get water as one of our final end products of this process. So here we see a picture here of what's going into this process. So we have 10 molecules of NADH going in, 2 of FADH2. Those are our electron carriers providing electrons for the electron transport chain to harvest energy from. We have 6 molecules of O2 going into this process to be our final electron acceptors. And at the end, we're going to produce water coming from that oxygen, basically, as well as producing our electron carriers again, no longer in their reduced form. So we can see they're in their original form, the NAD and the FAD. So they're going to go and sort of cycle back to the beginning of this process so we can reuse them. So they don't get completely used up. They basically are regenerated by the end of this process. We're also going to get a ton of ATP. Don't get hung up on the specific number because this is variable depending on tissue type, but 28 would be a reasonable number, 30 would be a reasonable number. We're going to get a lot of ATP, something in that range produced here. Okay. So if we're looking at our mitochondrion, we're not going to talk about the names of the specific coenzymes or really any of the molecules here embedded in the membrane, but here you can see how things are getting passed along down this chain. That's what the chain is, this series of molecules embedded in the chain. So what's happening, basically, I think these will click through. Okay, yeah. So this is where we are in the mitochondria. So this is our inner membrane. So this is the space we're looking at. You can see this little inset. And so that's what we're looking at. And so what happens as the electron transport chain is running, we have our electron carriers come in, so our reduced NADH here. It passes its electrons and some hydrogen to this molecule here, this first molecule in our electron transport chain embedded in that inner membrane. And as we rip off these electrons, we basically use this to power the pushing of a hydrogen ion across the membrane. So the reason we see this high concentration of hydrogen ions in the intermembrane space, so they're not free in the cytosol, they're not able to float around the rest of the cell, they're still in the mitochondria, they're stuck between the 2 membranes, and this part here is basically the very center of the mitochondrion. So the reason we're able to go against our concentration gradient, which should require energy, is because we're using these 2 electrons to power it. So that's what's happening here. So we're using this redox reaction to take those electrons off the NADH, use it to power the increase in this hydrogen ion gradient. And so as we move down the chain, we continue to do that until we get to our final part of the electron transport chain, where those electrons bind on with a couple hydrogen, and our final molecule accepting those electrons is oxygen. So when the whole electron transport chain has run its course, what's left over binds onto oxygen and gives us water. Okay. At this point, we still have a concentration gradient. So we have this high concentration of hydrogen ions in the intermembrane space compared to the matrix. So the matrix is the center of our mitochondrion. And so what we're going to do is we're going to allow these hydrogen ions to flow through ATP synthase. So this is another molecule, technically an enzyme, but a very specialized one that spins as the hydrogen ions flow through it. And as it spins, it creates ATP. So this is the molecule that's actually responsible for the creation of the ATP. So we have the electron transport chain harvesting energy in our electron carriers to produce a hydrogen ion gradient. And then the gradient is released, allowing hydrogen ions to flow into the mitochondrial matrix. So they're not floating out of the mitochondria, they're flowing back into the center. And that releases the energy stored in this gradient in the form of ATP. So here is where we would have our little phosphate ions attached on to our ADP to produce ATP. This is really efficient. This is the best way to produce ATP. If you want to get a lot of bang for your buck, you want to get a lot of energy from your glucose molecule, this is the way to do it. But you need oxygen in this process because if you don't have this final electron acceptor, you can't run the chain. Okay. So if we wanted to step through all of cellular respiration with its constituent steps, we'd start outside the mitochondria. So we're out in the cytosol, out in the cytoplasm. We have glycolysis. So we feed in this oxidized form of our electron acceptor, NAD, get out NADH. So we're starting to produce these electron carriers that are going to go into this final process. We're also producing a little ATP out there in the cytosol. Okay. Then we have our linking step that moves us into the mitochondria. So we got 2 molecules of pyruvate at the end of glycolysis, move them into the mitochondria, produce a little bit more of this reduced electron carrier, produce a little bit of CO2, so we're starting to pull off some of our carbons. And we produce 2 acetyl-CoA molecules to continue on with the rest of the steps in this process, which continue with the Krebs cycle, which looks very complicated, but fundamentally, the biggest goal of the Krebs cycle is to produce a lot of these reduced electron carriers. They're going to go to our final step, which is our efficient way of producing ATP. But we also have some CO2 being produced here. That's the rest of our carbon dioxide from the glucose molecule. You can see 4 plus 2 is 6. That's how many carbons there are in a glucose molecule. We're also producing some ATP. So note the numbers on these slides here are telling you per glucose molecule. So when you see 2 ATP here, that is telling you after 2 rounds of the Krebs cycle, because we had 2 molecules of acetyl-CoA, because we had 2 molecules of pyruvate. So that's why the numbers here differ than when we were looking at just 1 Krebs cycle before. Okay. So finally, oxidative phosphorylation happens using the electron transport chain. So here's where we need our oxygen. All our reduced electron carriers go back to their original forms because we peeled off the hydrogens, we created a gradient, we used up the energy stored in those electrons, we shoved that hydrogen ion gradient through ATP synthase, it spun around, gave us a ton of ATP, and all we're left with to go out into your bloodstream basically is water. So we have our full equation if we want to draw it out with the parts that make the ATP. These do kind of cancel each other out, which is why they're not usually in there. But we put in ADP, which had 2 phosphate groups, plus our extra phosphate groups that we're going to tack on, end up with our ATP. Now, as we can see here, the number of ATP molecules produced is variable. Okay. So without oxygen, we do still need to be able to break down glucose. And this is because sometimes your body uses up oxygen like basically too fast. If you're exercising really hard for a long time, you're trying to produce a ton of ATP really quickly. And at some point, you're using up oxygen more quickly than you can get it into those tissues. So you're breathing fast, right? You're trying to get in a lot of oxygen, but it still takes some time for it to get into your bloodstream, get into your cells, and you don't want your cells to totally quit because they have no energy at that point. So in situations with low oxygen availability, the electron transport chain starts to back up. So it starts to back up because we don't have that oxygen to accept those electrons at the end to give us our water molecules. So we're basically stuck in this state where the electron transport chain has started, but we can't release our hydrogen ion gradient. So when we back up the electron transport chain, it basically backs everything up. So the fact that we can't send stuff to the electron transport chain actually also even has effects further back in the process. So it also stops the Krebs cycle. Basically, it's going to stop what's going on inside the mitochondria. So the problem is we can still use glycolysis to make ATP, and that is what we're going to do. But glycolysis requires the input of NAD, which is the oxidized form of this electron carrier. So NADH is a reduced form. Usually the electron transport chain pulls off that hydrogen, uses up an electron, and we're good. We can send that NAD back to the beginning of the process. But if we don't have that electron transport chain running, this process isn't happening. So we don't have that NAD to feed into glycolysis. So your body comes up with another way to turn NADH into NAD so that we can continue to run glycolysis, produce a little bit of ATP, even though it's not that efficient. And the way we do that is we synthesize lactate. So yeah, so in this image, you can see our glycolysis happening out in the cytosol the way it normally would. So we're putting in glucose, we're getting pyruvate, we're using NAD in this process. That's just part of how glycolysis works in those 10 steps. But because our electron transport chain isn't running, because we don't have oxygen, we end up with buildup of NADH, which we need to get back into that NAD form so that we can continue glycolysis. So instead of taking that pyruvate, putting it into the mitochondria, starting the Krebs cycle, what we do instead is we take that pyruvate molecule, we use an enzyme lactate dehydrogenase to form lactic acid. So when you hear that people's muscles burn because of a buildup of lactic acid, what we're talking about is this process, is the fact that we're taking pyruvate, breaking it down to lactate or lactic acid in order to get this NAD to continue to power glycolysis. That's why this is happening. This is like a workaround because we don't have our normal process running to form this molecule for glycolysis. Okay. So we're going to work now on a short case study where we will be applying our knowledge of cellular respiration. So we're going to think about these steps critically, and we're going to go through a problem of something that can go wrong, and we're going to try and identify which of these steps is causing our problem.